All right, guys, welcome to the urinary system PowerPoint. This is the last PowerPoint before your lecture exam number 5. And lecture exam number 5 is going to be on the 14th. So if we were in school in person, next week would be spring break, and then the following week would be lecture exam number 5. So you do have about a week and a half to study for this next test, and it includes 3 PowerPoints, and it's the last 3 that we've covered. So to finish off this module, the urinary system is a system that makes urine. It involves a few different organs, so we'll talk about the functions first, and then the macro anatomy, and then the micro anatomy. So functions of the urinary system, first and foremost, is to maintain your blood chemicals. We need to make sure all the salts, all the protons are regulated in your blood to make sure that we're not too concentrated or we're not too acidic. The second job is to get rid of toxins that are in your blood, things like creatinine, ammonium, anything that's nitrogenous based needs to be excreted because those guys can be toxic to your body. So 3 main components of waste, again, creatinine, urea, and uric acid. So these are the 3 main ones that you're getting rid of. Now, the urinary system is made out of 4 major organs. So you've got the kidneys. They connect down using the ureters to the bladder. And notice the bladder is covered with a smooth muscle. And then the bladder excretes the urine out of the urethra. Males and females have the exact same anatomy, except the male urethra is substantially longer than the female urethra. And we'll talk about that at the end of this PowerPoint. This is a great illustration, by the way, to put on your lecture exam. This is also a great illustration to put on your lecture exam, especially because now we won't be able to dissect a kidney. This is what the kidney dissection would have looked like had we done it in class. It looks much better in this illustration than what you would have seen, so you're not really missing out on a whole lot. The kidney structure is made out of a membrane that covers the entire kidney called the fibrous capsule. Remember, anything that's fibrous is fiberful, and it's a pretty tough membrane. The inside of the kidney is called the hilum, and then we have this outer region, this light area. This is called the renal cortex. The inner region, where you see all these dark red triangles, this is called the renal medulla. The cortex is going to be the outside of the kidney, and the medulla is going to be the inside of the kidney. In the medulla, you see these triangular structures, and these triangular structures are called renal pyramids. You're going to be filtering out the blood within these pyramids here. And then inside the middle, you have the renal pelvis. This is where you're collecting all the urine and sending the urine out of the ureter. So right here is where you're going to have the renal pelvis collecting the urine, again, sending it out of the ureter. And then the other 2, you can clearly tell that there is an artery and a vein. The artery brings in blood to be filtered. The vein takes out the blood that's going to be kept in the body, while the ureter takes out all the excreted toxins. Now, this is a very, very important slide. Please star this slide everywhere. This is going to be huge on the lab practical. Almost all of these labels will be on there. So be sure that you know this picture. This picture is the same as the last picture, except now you see continuations of the artery and the veins. So where we're going to start is at the artery. I'm going to tell you the names of every time you branch out. Each branching has a different name. So the renal artery branches into segments we call segmental arteries. The segmental arteries enter in between these lobes here, 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 there, and there. Those are called interlobar arteries. Again, interlobar arteries because it's between the lobe. From the interlobar artery, they kind of sit flat at the bottom of these pyramids here, right there. You see one right there. You see one right there. Those are called your arcuate artery. The way I remember that is because it's an artery that arcs over. So if you follow the interlobar one, it arcs over. From there, you have these little rays of sunshine that go to the cortex of the kidney. These are called cortical radiate arteries. We call them that because they're in the cortex and they're radiating out like sunshine. So cortical radiate arteries. If you follow the same pathway back, you'll have a cortical radiate vein, the arcuate vein, you have the interlobar vein. The only thing you don't have here is the segmental vein. This is simply called an interlobar vein all the way until you get to your renal vein. So everything is the same in arteries and the veins, except you do not have a segmental vein like you do the artery. Again, another picture. This looks different, but hey guys, look, it's the same thing. Renal artery to the segmental artery to the interlobar artery to the arcuate one, then the cortical radiate one. Once you get to the cortical radiate artery, and I'm going to go back a slide here, each of these little branches that you see that are little rays of sunshine, each of those connects to a structure called a nephron. That's where we're going to spend most of our time learning the anatomy. They're not shown on this picture because they're far too small, but that's where you have the dotted lines surrounding it. So these structures are all structures of the nephron. So blood goes in, filters in the nephron to get rid of all the waste, and then anything that's going to stay in your body goes back out the veins. Anything that's going to be peed out is going to stay inside the nephron and go eventually to your bladder. We will see this whole structure in just a moment. So from the cortical radiate artery, again, we're going to enter into a structure called a nephron. The nephron is shown right over here. So I'm going to go over this picture first, and then I'll go back here so we can go over the names once again. So here's your cortical radiate artery. This is it right here. A little ray of sunshine, this little branch comes off, and you call this an afferent glomerular arteriole. We call it afferent because it's coming towards this loopy structure. This loopy structure, it's just a capillary bed, and it's called a glomerulus. The capillary bed, if you recall, is only a single cell layered thick. So what is easy to do across this capillary bed? Well, diffusion. So blood comes in, goes into this capillary bed, all the waste diffuses out, and it's a process called filtration. Anything that doesn't get filtered out as waste is going to enter back into your blood circulation through the efferent arteriole, which eventually turns into this system called peritubular capillaries. These capillaries will kind of wind around the nephron before going out and becoming a cortical vein. So again, from the beginning, the cortical radiate artery branches off into the afferent arteriole to enter the glomerulus to do filtration of all the dirty stuff in our blood. Anything that stays in the blood, so whatever you don't want to be filtering out and peeing out, goes back into the efferent arteriole through the peritubular capillaries so that it can combine and converge back to the cortical radiate vein, the arcuate vein, the interlobar vein, and remember again, we're skipping the segmental. We're going straight to the renal vein to go to the inferior vena cava, and that goes to the heart. So far we've just covered vasculature. So blood goes through the cortical radiate artery to the afferent arteriole to filter out of the glomerulus, and that filtrate goes into the nephron to eventually be peed out. And then you have the remaining solutes in blood staying in circulation, leaving out of the efferent arteriole. And the efferent arteriole turns into a capillary bed that runs alongside this nephron tubule. So the nephron tubule and the capillary bed, the peritubular capillary, run side by side until the end, where you finally release out the garbage as urine, and at the end you take the rest of the blood that's in here into the cortical radiate vein to go back into circulation. Now, one very important point I want to make on this slide is a few terms. 1, 2, and 3. The process of filtering the initial filtrate out of the blood, that's called glomerular filtration. The term is right here. Now, sometimes you filter out a lot more than just waste. You start filtering out good proteins, good salts, too much water that you don't want to be peeing out. So your tubule has a chance to say, hey, I filtered out a bit too much. Do you think we could reclaim some of this filtrate back into the blood? Because I don't want to pee it out. I need these proteins. I need this salt to stay in my body. So there's this way of passing solutes and water back and forth until we can balance out what truly is waste to pee out and what you should be keeping in your blood. So the process of moving solutes from the tubule into the blood is called tubular resorption. Another word for this that I'd like you to write down because I use it more often is tubular reabsorption. Again, tubular resorption is the same thing as tubular reabsorption. Then sometimes the filtrate doesn't completely filter out. Sometimes you keep waste in the blood, and the blood then says, oh no, I kept some waste in here. It didn't filter out when it was supposed to. Do I just have no choice but to keep it in my body? No, you do have a choice. You can take that waste and put it back into the tubule to get peed out. Taking any solutes and water from capillary into tubule is called tubular secretion. So a lot of good information on this slide. Make sure that you know the terms and the direction of flow of the blood through the capillaries and through the nephron. So again, here are the terms. You guys can read over them if you'd like. Filtration, resorption, aka reabsorption, and secretion. So I'm using the same picture, but right now we're just going to focus right over here. Let me switch to my pen. So this is the structure we're going to focus on right now. You already learned afferent arteriole, glomerulus, efferent arteriole. But this glomerulus is surrounded by capsule. The whole structure of glomerulus and capsule is called the renal corpuscle. So the renal corpuscle is this entire structure in the box. It's made up of the glomerulus, which is the capillary bed, and these capillaries are fenestrated. If you recall what this word means, it means it has holes. Yeah, it's very difficult to write on a MacBook. So this capillary has holes in it. That allows for the filtration. The capsule space around it is made out of two layers. This outer layer where my pen is drawing, this outer layer is called the parietal layer. Parietal means outer, and it's made of simple squamous epithelium. The inner layer, which you can't see on this picture, it lays right down on top of the glomerulus. That's called the visceral layer. You are going to see this in a very good picture coming up. This is made of podocytes. I need you to know this cell type. A podocyte is a cell that looks like this. It has like these little fingers. Cell body, there's your nucleus. So it's just a cell, but it's a weirdly shaped cell. These are the type of cells that are found on top of the glomerulus. And again, you're going to see this perfectly in a coming illustration. Before I read over this slide, I want to show you that illustration so you can understand what I'm talking about. So let's look at a close-up look at the renal corpuscle. The afferent arteriole brings in blood into the capillary bed. This capillary bed is called the glomerulus. Anything that doesn't filter out leaves out of the efferent arteriole. This glomerulus is going to be found inside a structure called the capsule. The outer layer of the capsule is called the parietal layer, and it's made out of simple squamous epithelium. You see that it's simple, one layer, squamous shape, so very flat, and it is epithelium. The inner layer, which is called the visceral layer, is made up of cells called podocytes. So here are the podocytes in yellow. You see the little legs and little fingers sticking out. These podocytes are the inner layer and they sit on top of the glomerulus. They're literally wrapped around the glomerulus. This is considered the inner layer of the capsule. In between the inner layer and outer layer, you have an open space called a capsular space. When blood comes in and filters out of the glomerulus, it filters out into this capsular space and it eventually all gets pushed into this opening right here. We haven't talked about what this is yet, but don't worry about it just now. Just know that blood comes in. It has to get filtered out of the capillary and in between all these little legs of the podocytes, and then it collects in the capsular space to go into the PCT. So I'm going to go back a slide now and just read over this. So that filtration out of the glomerulus, it's made up of 3 layers. The wall of the capillary bed, which we call a fenestrated endothelium. Remember the word fenestrated means holes. The basement membrane, which is like the glue that sits on top of the capillary bed. And on top of that thin layer of glue, you have the podocytes laying on top. The podocytes, and let me get a pen and draw this for you. Let's say you have one podocyte here, and then you have another podocyte here. Their legs do this kind of intertwining dance between each other, which leaves the space in the middle for filtration. Filtration slits are those spaces in between the legs of the podocytes. The tighter it is, the tighter filtration is going to be. So what is allowed to get through the fenestrations and filtration slits? Well, these guys here. You're going to allow passage of water because it's tiny, ions because they're tiny, glucose you're going to allow because it's relatively small and it's not charged, amino acids are tiny, and urea is waste. What you're not going to allow to filter out into the tubule is things like red blood cells and proteins. Here's how we think of it. If you go give a urine sample to the doctor, should you be finding red blood cells in your urine? What would your urine look like if you were peeing out red blood cells? It would look bloody. Is bloody urine normal? It is very abnormal. That is not something you want to see. Proteins as well. Although finding small amounts of proteins is okay in the urine, you don't want to be finding high amounts of it. So you shouldn't be filtering out too many proteins or red blood cells because these are way too big to get through these little tiny filtration slits or to get through fenestrations. But what can get through these guys are small molecules like this. So let's take a closer look at what this would look like. So here we go. I'm going to circle a few things that I think are important. All right, capillary bed, single cell layered thick, and they're fenestrated. Fenestrations are these little holes. Do you think a big Mongo red blood cell could fit through that? Probably not, because if I were to draw a red blood cell for you, no joke, it's literally that big. I'm not kidding. I'm not like exaggerating or anything. This is how big a red blood cell is. This red blood cell, there's no possible way it's going to get through that tiny little fenestration. So this is not something that should be peed out or filtered out of the glomerulus, eventually going into the urine. Sitting on top of that is the podocyte. These little openings in between the podocyte, these are called filtration slits. And filtration slits are those little openings. So both of these guys are your 2 layers of filtration. Now, what's not shown on here is the basement membrane. We don't usually show basement membranes on these illustrations because it's such a thin layer, and it really acts just like a glue to make these podocytes stick onto the wall of the capillary. So this is a great illustration. If we look at a cross-sectional view, what you see is on the left side is inside the capillary. You have your little pores, which are fenestrations. You got that thin basement membrane, And then you've got the legs of the podocytes here, leaving spaces in between called filtration slits. So it shows you that molecules, like maybe ions or salt, can get through this small pore and then get through the filtration slit. So this is where we kind of change the gears a little bit and continue out of the glomerulus. So if I can just, let me see if there's a picture. There's not. I'm going to draw this for you, and I highly suggest you drawing this as well. All right, so what I'm drawing here is a glomerulus. There is your glomerulus. The outer region is the capsule. And then we're going to draw a series of loops and lines, and I'm going to label them all for you and show you what each of them is. All right, I had to pause and redraw the illustration because my dog started barking and I had to delete it out. Anyways, here you see the entire nephron loop, and I'm going to redraw this glomerulus in here. So far we've gone over this section. The following sections we call the tubule. So this whole thing is called the renal corpuscle. These sections are called the renal tubule. The tubule is broken up into 1, 2, 3, and 4 sections. I'm just going to draw the lines and connect them. First section is called the PCT, the proximal convoluted tubule. The second section is called the loop of Henle. And the third section right here is called the DCT. And the last section is called the collecting duct. So PCT, loop of Henle, DCT, collecting duct. This loop of Henle, if you notice, it has a descending part to it and an ascending part to it. This is called the descending thin limb, and this is called the ascending limb. If you notice the ascending limb right here, it's broken up into 2 structures, the ascending thin limb and the ascending thick limb. You will see the differences in thickness on the loop of Henle in a coming illustration. And then we'll look at a few more of the other structures. Collecting duct, I do want to give you a little bit of physiology on this because this is a really important section. Again, collecting duct is that last section that we drew. The collecting duct is going to receive like the final filtered, balanced urine. And at the collecting duct is where you're going to reabsorb a ton of water. So this is where you're going to have a lot of water reabsorption here, meaning you're going to take water from the urine and pull it back into the body because your body says, hey, maybe we shouldn't be peeing out this much water. Water's really good for us. Let's reclaim it. So this is where water reabsorption takes place. So it's an important portion in conserving body fluids. Now, there is a hormone that affects the function of the collecting duct. And that hormone is going to be called ADH. I don't know if we've gone over this before. I feel like we have, but if not, let's go over it again. ADH is a hormone called anti-diuretic hormone. If you just break down this word, anti-diuretic hormone, it's a hormone that's against being a diuretic. What do diuretics normally make you do? Do they make you hold in pee, or do they make you pee out more water? So diuretics, their job is to make you excrete out more water. But if this hormone is called anti-diuretic hormone, it's against the excretion of water. That means this hormone, when your pituitary gland secretes it, tells the collecting duct, hey, don't pee out that much water. Let's reabsorb the water back into the body. So it essentially, ADH increases the permeability, that means the ability of water to pass through the collecting ducts and reabsorb more water. So if you secrete ADH, you're going to hold water in and pee out less water. This makes your urine volume output much smaller. So there are two classes of nephrons. So they both look the same, the structure that I drew on the slide 2 slides ago. And the difference is where they're found in the kidney. Now remember, the kidney has a cortex and a medulla. So if this is my kidney here, there's my ureter. I'm going to just draw an outline. It has a cortex, which is this guy here. I'll label it with a C. And then the middle of it is called the medulla. Label it with an M. Cortical nephrons are nephrons that are found in the cortex of the kidney. Juxtamedullary nephrons are found in the medulla of the kidney. The majority of your nephrons are cortical nephrons. Maybe 15% of them are juxtamedullary nephrons. And the reason that is because as you go deeper and deeper down into the core of the kidney, you have an increase of salt concentration. So I'm going to write the word salt here, and then I'm going to put it into brackets because brackets means concentration. So in the middle of the kidney, you have a very high salt concentration. On the cortex of the kidney, you don't have that high of a salt concentration. Because of the high salt concentration in the middle, it allows the juxtamedullary nephrons to reabsorb more water, making the urine a lot more concentrated. You do not need to know the mechanism or reasoning behind this. Just understand that any nephron that's in the middle of the kidney is going to reabsorb more water because of the high salt concentration in there. And this is what they look like. So the cortical nephron is going to be this guy here, found in the cortex. The juxtamedullary nephron is going to go deep into the medulla. Now, the structure that actually is different in them is the loop of Henle. Look at how short this loop of Henle is, staying within the cortical region. But look at how long this loop of Henle is. It goes deep down into the medulla. So that's their main difference, where they're located and the lengths of the loops of Henle. Again, the juxtamedullary nephron, because it goes deep into the core of the kidney, is surrounded by a lot more salt, which causes more reabsorption of water. Now we're going to take it back to the glomerulus, and I'm going to give you a little bit of the microanatomy here in terms of cell types, and I'm going to go ahead and use my pen to circle them. We got cell type 1 here. We've got another one surrounding the afferent arteriole, these dark red ones here. I'll circle both of them. We've got another one here, these lavender-looking cells, and then you've got these lavender-looking cells all inside the glomerulus. These 4 cell types will help the kidneys control how much they filter. And we're going to talk about why that is. So that whole, let me go back, that whole structure right here, this is called the JG complex. And let me draw this out. Usually I would draw this on a board for you. But what a nephron actually looks like is a little bit different than what we've taught you so far. We teach you in a basic form to understand the different structures of a nephron, like PCT, loop of Henle, and DCT. But in real life, you have the PCT, you've got the loop of Henle, but the loop of Henle does this, whoa, wraps backward and comes right in between the afferent and efferent arteriole to turn into the DCT, then into the collecting duct. This is the structure of a real life nephron. This region right here, where the top of the collecting duct turns into the DCT, this is going to be what we're looking at here. This opening here is the top of the collecting duct turning into the DCT. The whole structure together is called the Juxtaglomerular complex, abbreviated as JG complex. So with that being said, I'm going to give you the names of all 4 of these cells, and I'm going to tell you their functions. It is your job to remember the names and remember their functions. So the JG complex altogether is a complex of interactions of the nephron with different cells that controls the filtration rate. So it controls how much you filter out of the blood. Cell number 1. These are called granular cells. If I go back a slide, there are these dark red ones here. These granular cells, I want you to write another name for them also. They are called JG cells. These granular cells, the JG cells, they're smooth muscle cells. So just like any artery, they're covered with smooth muscle, right? So these smooth muscle cells are called JG cells or granular cells, and their job is to constrict or dilate the afferent arteriole. So let me give you just an example. What would happen to the amount of blood coming into the glomerulus if these JG cells constricted? Think about it for a moment. If these JG cells contracted and constricted the afferent arteriole, do you have more or less blood coming in? If you said less blood, you would be correct. And if less blood gets into the glomerulus, do you think you're filtering out more or less waste? You are filtering out less waste. So filtration rate goes down. Same thing if you dilate this afferent arteriole, you open it up, a lot more blood gets in, and that generates really high pressure in here because you have now a lot of blood in this little area. It's going to push more waste out, and that's going to increase your filtration rate, essentially increasing how much you're filtering out. So these granular cells, one of their big jobs is to constrict and dilate the afferent arteriole. That's not written here, so I want you to write that down. A second big job is that granular cells, aka JG cells, secrete renin. If you could do me a favor, cross out the word hormone and replace it with the word enzyme. Renin is an enzyme, not a hormone. So the JG cells secrete renin, but not all the time. It only secretes renin when your blood pressure drops. So when your body senses that your blood pressure has dropped, your JG cells are going to make renin, and renin is going to help your kidneys filter more efficiently in response to lower blood pressure. So blood pressure is kind of a big deal in terms of kidney function. If I go back a slide, if the blood entering the glomerulus was at a really high pressure, is your filtration rate going to go up or is it going to go down? If blood is coming in at a very high pressure. So if you have high pressure blood, it's going to filter a lot. But let's say you were someone who had chronically low blood pressure, or for some reason you were bleeding out, your blood pressure was dropping for whatever reason. If you have very low blood pressure, you're not going to have high pressure blood coming into the glomerulus. It's going to be low pressure. And if the blood coming into the glomerulus is low pressure, do you think you're going to be filtering out a lot of waste? Or do you think the waste is just going to stay in the low pressure blood? The waste is going to stay in the low pressure blood. So with someone who has low blood pressure, they're really sending in blood and it's not getting filtered out effectively. So their kidneys have a backup way, and the backup way is to secrete renin. And renin helps the kidneys increase their filtration rate. You don't need to know how it does it right now. You just need to know that renin is secreted when your blood pressure drops, and it causes the kidneys to regulate that filtration rate to make sure you're not holding in too much waste in your body. Then you have macula densa cells. So I'm going to go back again. These are your macula densa cells. These cells have a ton of little receptors that hang off of them like little hairs. Those receptors are constantly monitoring the concentration of fluid that's coming in through here. So these macula densa cells can say, hey, there's way too much salt in here. Lower your filtration. Or the macula densa cells can say, hey, there's not enough salt and there's too much water. can you guys increase your filtration to get more salt and waste out? So these macula densa cells will monitor the concentration of the filtrate, and they will send signals to the rest of the cells, alerting them when filtration is too high or when filtration is too low. It says here that the macula densa cells can communicate with the JG cells to secrete renin. This renin, that enzyme, remember, is going to help you with your blood pressure, and it's going to help you with your filtration. And you only secrete renin when your blood pressure drops, which means your filtration is also lower. Mesangial cells and extraglomerular mesangial cells have somewhat the same name, but they're different. These guys here, the purple ones, are called mesangial cells. The ones out here in this triangle are called extraglomerular mesangial cells. These guys and these guys talk to each other. The macula densa cells can tell the extraglomerular mesangial cells to communicate with the mesangial cells in the glomerulus to constrict or relax. These mesangial cells have the ability to constrict this capillary bed and lower filtration rate, or dilate this capillary bed and increase the filtration rate. So the mesangial cells can regulate blood flow in the glomerulus, and then the extraglomerular mesangial cells communicate with the macula densa cells. And again, this picture is coming up. It's a great picture. If I wanted to put this on a lab practical, I could put it on there and ask you to label the afferent arterial, the efferent arterial, and the 4 cell types that you just learned. So once you filter out of the nephron, you collect that urine into the pelvis of the kidney, and then you send that urine out of the ureters into the bladder. The bladder is going to be your main storage facility of urine. It can stretch out, it can coil back in. It just depends on how full it is. The urinary bladder is covered with smooth muscle, which is why it's able to stretch out and relax. And the full bladder can get so big sometimes that it can stretch out from the pelvic cavity up into the abdominal cavity, which is why your tummy kind of looks big and swollen when you have to pee really bad. And then the last thing is from the bladder out to the external environment is the urethra. Male urethras are about 20 centimeters. Female urethras are about 3 to 4 centimeters. Now, what controls when urine is allowed out of the urethra? Well, you might think, well, I'm an adult. I control when I pee and when I don't. But babies and young children don't have that control consciously yet. That is because there are 2 urethral sphincters. There is an internal one that is made of smooth muscle, which means it is involuntary, but it is this external one that's made out of skeletal muscle, and it is in conscious control. This is the sphincter you are trying to control when you're potty training a little kid. So the internal sphincter is controlled by your body itself. The external sphincter is controlled voluntarily with conscious control, which means smooth muscle here and then skeletal muscle here. So some disorders of the urinary system, UTIs. These are very common in females because the female urethra is so short. It's not a very long distance for bacteria to make its way up to the bladder. For males, the bacteria would need to go to about 20 centimeters before making it to the bladder, and so they don't get them very often. Renal calculi, this is a fancy schmancy name for kidney stones. Bladder cancer, more men than women have this. And then kidney cancer. This usually is caused by overgrowth of those tubules we talked about. So the tubules that are found in the kidneys, when the epithelial cells start to multiply rapidly, number one, too much growth can affect filtration. So you're going to find that you have weird colored urine, weird smelling urine. Maybe you're not urinating at all, and that could indicate kidney cancer. All right, so that's the urinary system, and that's going to be the last lecture before you take your next test. The next test will not be open note or open book like the last one was. The next test will be using Proctorio in a much stricter fashion than you used it on the previous exam. So be sure that you're studying this material. And remember, it's only 3 PowerPoints. And be sure to email me anytime you want. I'm just home, as all of you should be as well, recording these PowerPoints and waiting to see if you guys get office hours. So let me know how it goes.